Alright, so we are going to, this is what we're basically doing. I told you that if I were you, this crazy thing, which we will come back to a lot, it is definitely not the place to start, I guess, to build your understanding of what's going on with cellular respiration. And so we had an extra glue in that we did here, and this is the bare basics of cellular respiration. It goes in, where does it go, what's it transformed into, what do you need, and we moved it down here. And we spoke a lot about, okay, so you say that you're going to have ATP that you've got to use, which means it's got to go into this part, which is glycolysis. When you use it, what do you end up with? So we built this whole piece here. So this is where I told you I would spend the majority of my time. So let's go back real quick and talk about the semester exam. So in the middle of this unit called energy, which is about cellular respiration and photosynthesis, we stopped at cellular respiration and we took a semester exam. So it may have left you with the feeling that you didn't have to know these processes in much detail, because the semester exam was not the unit exam. The semester exam equally gave weight to everything you did. The next test you take will be the exam on energy, which will focus primarily on cellular respiration and photosynthesis. And as always, those tests are half new stuff, so half of the exam will be the very detailed level of cellular respiration and photosynthesis, and the other half will be all the other stuff we've done, the old material. So that next test won't feel at all like the semester exam. Most of you who took it said it wasn't nearly as difficult as you thought it would be, which is wonderful because the questions are, I'd say almost all of them were AP level questions. So I think the difference was you had an equal amount of things. It wasn't heavily weighted toward one topic like a unit exam. But that's coming up, and so we want to be sure that we can actually understand cellular respiration mostly from an input-output requirement level, because a lot of the questions are, hey, what if you are unable to oxidize NADH? What's that going to do? What would be affected and why? So you want to be able to clearly understand, here's what I have to have in order for this to start. When it's over with, here's what I'll have as end products, and then know where all those go. So I thought, go to your next clean page. I just turned mine this way so I could have a little bit more space. And I've counted over 7 just to chunk it into equal spaces. And then I had about 6 inches worth of space here. So I've marked every 1.5 inches. So at 1.5, 3, and 4.5. And that'll cut these into 4 fairly equal spaces to write. So here's this piece. I'm going to put the processes here. So the very first column will be the steps in cellular respiration. So we'll start off with glycolysis, go through pyruvate oxidation, Krebs, and electron transport chain. And then over here, I'm going to put what are the inputs into this system, because this would be a system in and of itself, and it would also be part of an entire bigger system. So what do you have to start? When you're done, what are you going to end up with? And then this is just going to be things that you need to know that are like extra. So this is just good to know. Okay, so I'll start off by just numbering what my steps are. So cellular respiration starts off by snapping glucose in half. That's glycolysis. Then you take those 2 3-carbon sugars that you created, pyruvates, through glycolysis, and you oxidize those. That just means you go through some type of breaking of bonds. So this is pyruvate oxidation. Now this particular step, sometimes they don't name it at all. They'll lump it into along with Krebs, or they'll lump it along with glycolysis, or they'll just skip it. But it is definitely another regulated step. And then we have, I usually just go with Krebs, but it can also be called the tricarboxylic acid cycle. Trying to think what else that they call it. I think that's pretty much the main one. And then here you have the electron transport chain. So you start off with glucose, and you fully metabolize glucose, oxidate it by the time you're through with Krebs, and then the electrons that you have gathered, you run them through the electron transport chain, and you go through oxidative phosphorylation, and you make a ton of ATP. So let's just walk through here with what goes in and what do we need. I think the easiest way to do that is I'll be doing a lot of flipping, but I'm going to go back here and we'll just make this a lot less busy. So we started off with glucose, right? So if we just do where are the carbons? In glycolysis, we start off with a 6-carbon sugar, which is glucose, and when we're through with glycolysis, you end up with 2 3-carbon pyruvates. So let's put that in there. So you start off with glucose, and we would have 1, and you'd end up with, following along with carbons, these glucose molecules get broken in half, and you end up with 2 pyruvate molecules. Okay, so that's the carbon piece. In order to keep glucose from moving back out of this GLUT1 transport protein that's in the cell membrane, we claim it, we phosphorylate it. That means it can't move anymore according to concentration gradient. It's now stuck inside the cell in this orange part, which is the cytoplasm. That activates or energizes this already not very energetically stable. The bonds in glucose are not as stable as they are in pyruvate, and we make them less stable with this phosphate group being added, and that would cost us an ATP. Then you've got this go, no-go step, where you're going to add this second phosphate group, which is fructophosphokinase. If we add this second phosphate group, we will split glucose into glyceraldehyde 3-phosphate and then into pyruvate. So this is highly regulated. We have lots of notes about that through allosteric regulation and feedback inhibition. But that's not what we're doing with these notes. Okay, all of those details are in the notes other places. This is a summary table. So it also, besides needing glucose, I need 2 ATPs as an input. I have to use them in glycolysis. So I have to use 2 ATPs. Overall, when you're through with glycolysis, you have 2 pyruvate. Here we are. For each one of these glyceraldehyde 3-phosphates that you create, because we've got 2 of them, as they're converted to pyruvate, you generate 2 ATP per G3P, and then you've had electrons because of this configuration between glyceraldehyde 3-phosphate and pyruvate. Pyruvate's more energetically stable, so there's a loss of energy, and it's going to be, now you used that to make ATP, 2 of them through substrate level phosphorylation right there in the moment, and you had electrons that came from this energy change, this conformational change, and so you formed an NADH. 1 per pyruvate, 2 ATP per pyruvate. So we'll come over here and put, so we had 2 ATP for each pyruvate. So that's going to be, I'm going to skip a space, and I'm going to write it right here. I'll create 4 ATP, and then I also created 2 NADH through the electrons from this bond breaking. All right, so that's the simplified, what do you have to start glycolysis? Glucose and ATP. When you're through with glycolysis, what are you going to end up with? Pyruvate, ATP, NADH. So these pyruvate are going to start, so they come over here, and they're going to begin the next part of cellular respiration. So you're going to have 2 pyruvate. So let's go here. Here they are. Here's the 2 pyruvate, 1, 2. In the presence of oxygen, this pyruvate transport protein that's in the outer membrane of the mitochondria is gated, and so when there's oxygen present, pyruvate can move in, and it moves in according to that whole gated piece with if you have oxygen or not. So in the presence of oxygen, pyruvate moves into this intermembrane space, which no work is done there. It's like a closet that we store hydrogen ions. Pyruvate will then be symported into this matrix area, and the way that works is you've got hydrogen ions at a very high concentration purposefully in this intermembrane space, low in the matrix. So they will naturally move according to their concentration gradient from the intermembrane space into the matrix. Pyruvate, however, will not. There's a low concentration of pyruvate here and a high concentration in the matrix. So a symport is going to use that momentum, that energy that the hydrogen ion has as it moves physically into the matrix according to its concentration gradient, and it takes that energy and allows pyruvate to move against its concentration gradient. So yes, this is active transport, but it's secondary active transport because you don't use ATP, you use a gradient. So pyruvate is then moved against its gradient into the matrix. So we start with pyruvate. Now we start snapping the carbons off. We're going to take a 3-carbon pyruvate, remove 1 carbon, and it's going to be removed as carbon dioxide, which will just go ahead and it can move according to concentration gradient, but it has no problem because it's nonpolar, passing and diffusing through any of these membranes. So it'll just move on out of the mitochondria, it'll move all the way through cytoplasm, it'll move out of the cell membrane and into blood vessels to be expelled as a waste product. So we have carbon dioxide that we created, 1 per pyruvate. That leaves you with a 2-carbon molecule, which is acetyl, and we need to add a CoA, so coenzyme A, to each acetyl for it to go to Krebs. When we broke this bond and snapped this carbon off, we had electrons, and those electrons were picked up by NAD and it was reduced to NADH. So we'll put that in there. So we started with 2-pyruvate, and what you end up with is 2-acetyl, 2-carbon dioxide, 2-NADH. So you end up with 2-acetyl and 2-carbon dioxide, and your electrons are carried by NAD, which is going to be reduced to NADH. We had to put CoA here, right? So we have to have available 2 coenzyme A in order to make acetyl CoA. So the acetyl CoAs are then going to move to the next step, which is Krebs, which is just like a garbage disposal for carbon. You throw any 2-carbon molecule in there and it just grinds it up, and you end up with the carbons all being released as carbon dioxide and your electrons being given to electron carriers. So we start off Krebs with 2 acetyl CoAs. There you go. And when you're done, you have no more carbons. Glucose is completely oxidized. You're done. So Krebs, you drop the acetyl-CoA into your little garbage disposal. The CoA is immediately recycled. It's going to go and be used again over here. You can see that, but over here for pyruvate oxidation, because we're still in the matrix. And these 2 carbons are going to go through this transformation of being added to a 4-carbon molecule, oxaloacetate, and then you're going to start a 6-carbon cycle where you snap off 1 carbon of the 2, another carbon of the 2, you're back to a 4-carbon molecule, and then you regenerate this precursor, this beginning part of Krebs, which is oxaloacetate. So as this happens, these 2 carbons are completely gone. They've been released as carbon dioxide as waste. So come over here and you're going to have 2 carbon dioxides per acetyl. I'm going to change that because we've been doing cumulative. So you'll have 4 total, because this is going to have to turn for 1 acetyl-CoA once, another acetyl-CoA twice. So you'll end up with 4 carbon dioxides total, which we did this down here if you want to look for our notes. So you have, for each turn, you got your 2 carbons as carbon dioxide. Your electrons went 3 to NAD for NADH, 1 to FAD for 1-FADH2, and then you did substrate phosphorylation and you created a GTP. So in total, for 2 turns, you're going to have 6 NADH, 2 FADH2, and then if you go back over here, you can't see it, but you had 2 GTP that were immediately converted to 2 ATP. So Krebs is just a 2-carbon garbage disposal. You plopped in 2 carbon. The acetyl-CoA, it gets broken off, so we'll have, and I'm going to kind of put it right here, 2 CoA, because they're able to go back up here and be used again in pyruvate oxidation. So you use them as the way to get into Krebs. You release them so they're not used up. They're recycled through these two processes. So your 2 carbons turn twice, you get 4 carbon dioxides, you get a bunch of electrons, 6 NADH, 2 FADH2, and then you do a little bit of substrate level phosphorylation and make a few ATP. You're completely done now. There are no more carbons, glucose is gone. All you have now are your electron carriers here. So let's count what you've got. You've got 2, 4, 10 NADH that are going to take their electrons and drop them off here at complex 1. And for truthful, and give this in a much more accurate statement, your coenzyme Q here, ubiquinol, is going to, because it's hydrophobic and can travel, it steals the electrons because it's more highly electronegative. And so as NADH approaches 1, Q will steal 2 electrons, and then C steals them from Q, and then oxygen steals them from C. So that's the true path of electrons. It's not this sweet handing them over. Each one of these is a bigger electron bully than the others. They're more electronegative, so they steal as they go. The FADH2s come to this not hydrogen ion pump to the complex, and Q can also steal electrons from FADH2 because it's got freedom of movement within this phospholipid bilayer because it's hydrophobic. So once that the electrons have been stolen from these electron carriers, you're going to also require oxygen, because oxygen is going to be what drives this electron movement. So let me go down here and put my 2 FADH2, which I got here. So now we've accounted for all the electrons to start the electron transport chain. I have to have oxygen, or it won't work. And when I'm done, I end up with ATP. Okay? So that's kind of the process of accounting for, in a very simplistic way, I'm looking for this. So in a very simplistic way, everything I have in blue came from that drawing. So now I need to think about, what do you have to have if you're going to create ATP in glycolysis? What would the pieces that you'd make that from, what would those be? So I'm going to use a different color, and I'm going to go back in red, and I'm going to think about, let's start here. So if you're going to make ATP, ATP is created from ADP, right? So it's only got 2 phosphates associated with it, and you'd have to have PI, which is inorganic phosphate groups. And if we're going to keep up with numbers, to make 4 ATP, you would need 4 ADP. And I always think of it in this particular way. This is your empty bottle, right? You've got to have that, because when you fill it, it's going to be this filled bottle. So if NADH is the full bottle, because NADH is reduced, it has 2 electrons on board. How did we make NADH? What would we have to have in order to make NADH? You have to have the empties, which are NAD. You'd have to have 2 of those. We don't always, like, we don't put hydrogen ions. They're very available. But in order to actually get NADH, you have to use NAD. NAD is the oxidized form, it's the empty. It's going to gain 2 electrons and a hydrogen, and it's going to become reduced, because that's what gaining electrons is. So it gains 2 electrons and forms NADH. So in order to create ATP, you have to have ADP available. In order to create NADH, you have to have NAD. So let's go down here and fix all of these NADHs and what you would have to have. Okay. So just like NADH is created from NAD gaining electrons, FADH2 is created from FAD gaining electrons. So I would need 2 of the empties in order to create the reduced FADH2. We said to make ATP, you need ADP. To make GTP, you would need GDP, right? Same thing here when you give that energy over and let it make real quickly a more commonly spent form of energy for the cell, which is ATP. So I would have to have 2 ADP available. They both need inorganic phosphate, because that's where the third one comes from, in order to make those. So here I would need ADP, right, to make the ATP that you're going to get from the electron transport chain. What happens when you run these NADHs through the electron transport chain? They give up their electrons. They're oxidized. So they're going to, like if this is the full of electrons, they would empty, because they gave the electrons to the electron transport chain. You create 10 NADs, so you empty these back up. That means they're free to go back up here to these 3 processes, here, here, and here, to be used to collect the electrons that are being made as we break bonds, and to put those electrons with these electron carriers. So we reduce these to make these full containers. So you would also empty the electrons out of the FADH2, forming 2 FAD. And then when your oxygen picks up electrons, believe it or not, that's reducing oxygen, right? Gaining electrons. It forms water, which is reduced oxygen. So you see that there here is, we're taking electrons and using these empties, these oxidized forms of the electron carriers, and in the first 3 steps you're reducing all of those electron carriers. They all go to the electron transport chain, they give up their electrons, so they're oxidized, and then we take those electrons away from these reduced forms of electron carriers, and we now are forming the oxidized form again. So what am I still needing here? So for carbon dioxide, here for carbon dioxide, these are going to be waste, and usually what we do is we just show that they're going to diffuse out. So glucose has 6 carbons. We account for the carbons. There's 2 here, 4 more here, so there's your 6 carbons total. And so those are gone and they're waste. And is there anything else we need to We accounted for our CoAs here. Here we would have to have, right, the 2 CoAs come out. We accounted for that. I think we got it all. Let me look over here and make sure that I don't see anything any different. So, yep, that's it. Oh, I know what it was, right here. When you use ATP and you snap off one of the phosphate groups and use that energy from that high energy phosphate on the third, you're going to generate 2 ADP and that inorganic phosphate group. So now I think it's all complete as far as what goes in, what goes out. So here's the point of it. It's really simple to say that, let's go here, that I'm going to, when I'm done, I'm going to make NADH from glycolysis, pyruvate oxidation, and Krebs. You're not, unless you have NAD available. So you cannot just make NADH out of nothing. There are precursor molecules that have to be available, which would be in this case the form which is NAD. And so the electrons, when they're gathered by NAD, so NAD is reduced and you form NADH, that's the only way you're making it, is if you have these molecules that go back and forth and shuttle between these 2 forms. So NAD has no electrons, NADH has electrons. And this molecule goes back and forth to shuttle, shuttle, shuttle between don't have any electrons, got some electrons. Don't have any electrons, got some electrons. Same thing here with ATP. I have stored potential energy. If I release it, I have ADP. If I gain energy enough to make this to where this conformational change occurs, and you can have the phosphate group, and it can be stored, then it's going to be in ATP. So let's think about what's good to know about glycolysis. Number one thing, it happens whether or not there's oxygen or not. That means it's anaerobic. That's huge. That is a good indicator that this is an extremely old process. Like it happened when Earth conditions were prior to a lot of photosynthesis, so really early when we didn't have a lot of oxygen in the atmosphere. Old, old, old. The other thing that would be good to know is it's in the cytoplasm. This is another piece of evidence that it's old. All cells don't have mitochondria, but all cells do have cytoplasm. So if this is really, really old and it could occur in all cells, it's going to be in a location where all cells have, like a location all cells have, which is the cytoplasm. Pyruvate oxidation and Krebs, those are aerobic, so they will not occur unless you have oxygen present. So pyruvate oxidation won't work unless you have oxygen to gate that transport protein for pyruvate to come into the matrix of the mitochondria. So this is located in the matrix. The same thing is true. You have to have oxygen for Krebs to work, and it occurs in the matrix. For sure, absolutely, you have to have oxygen as the final electron acceptor for the electron transport chain. So it's aerobic. In the electron transport chain, go back to your picture. So we drew a great big one in this inner membrane of the mitochondria, and then we just showed ourselves that there's gobs of them. Gobs. And that's why this membrane is folded, because it increases surface area. If you think about it, if you took a piece of paper, and here it is, and then space-wise, if you put a lot of folds in it, I can now fit that paper, same paper, amount of surface area in a much smaller space. So you can cram a lot more. So increase in surface area gives you more. So electron transport chain is in the inner mitochondrial membrane. And they're localized there because there's a sequential order. And so putting these complexes, which are, they're proton pumps, but they're protein complexes. They're transport proteins. Really, there are more than one to each one, but it doesn't matter. That's what they are. They're transport protein. So they're integrated. They're integral. They are transmembrane. 1, 3, and 4 are, because they're a pump. And they're associated with each other, localized in this membrane. So that's how you can keep them together, because you've got, like, ubiquinol can move freely between 1 and 3. C's hydrophilic, and it's associated with this outer face of this inner membrane over here toward this inner membrane space, but it moves and shuttles between 3 and 4, and then oxygen can snatch its electrons. So all of these have to be in order, and then you get, ATP made like crazy as you start releasing this hydrogen ion gradient. Okay, so here, these 3 pieces are about making electrons. So we'll put here that electrons are going to be released, and they're going to go to electron carriers. So, whoops, that's crazy numbering. So your electrons are released here. In electron transport chain, you use those electrons that were released. So by the time you get to the Krebs cycle, you have fully oxidized glucose. All gone. So the 6 carbons that you had, you've broken them into pieces of 2 carbon dioxides in pyruvate oxidation and 4 carbon dioxides in Krebs, and they're gone. All you've got when you get here, like this part right through here, is, hey, I've got all these electrons that I've collected. Oh, okay, bring them here and let's run them through. And then we'll use them to build the hydrogen ion gradient, and we'll let ATP synthase make gobs of ATP. So these are not about ATP. Okay, these are about, this is about electrons, electrons, electrons. This process is about ATP. So glycolysis, pyruvate oxidation, and Krebs are about getting the electrons out of glycolysis so that you can save them in electron carriers to take to the electron transport chain, and then let those electrons be moved. And if you look over here in our little figure, the electron starts off, see the height, way too high of energy for the cell to use. So if you think of it like toothpaste, we got a full thing of toothpaste. Nobody brushes their mouth, their teeth with a full thing of toothpaste. You just squeeze it out, squeeze it out, squeeze it out, squeeze it out. And that's what we do. Every time that we squeeze the electron, when it goes to a different electron carrier, you drop the electron's energy, and that energy is used to pump hydrogen ions across the inner membrane and create this hydrogen ion gradient. So this electron's high energy, it drops in energy when Q holds it tighter. That energy is used by complex 1 to pump hydrogen ions. As C steals the electrons from Q and holds the electrons tighter, that energy that the electron dropped from being held tighter is used to pump hydrogen ions across complex 3 into this inner membrane space. And then finally, when oxygen is the final electron acceptor steals the electrons from C, that drop in electron energy is used to pump hydrogen ions for complex 4. And so you drop energy, drop energy, drop energy, and you use that to power your pumps. All right? Maybe something you have to watch more than once. So that sums up the whole unit, but the details are a lot. So I'm getting close to my budget for this video. So I think what I'll do is I will go back all the way probably to here and just walk you through these notes again. I did that in a study video. I kind of feel like that I'm doing this a lot, but hey, it's a new semester, and we're going to talk through what are the important things in here that you just got to understand. I'll do that in the next video. But I would study this, and I would think about a test question that might say, in a cell, and I'm just making this up, a chemical is added that will not allow NADH to be oxidized back to NAD. Like it stays in this form. It's like it's broken. What parts of cellular respiration would be affected, and then why? So if you think about it, glycolysis would be affected because you need to have NAD, and you just told me that NADH is broken. Like I can't recycle it back to NAD. So eventually I'm going to run out of NAD. I've got both in my system, and I can work in this direction, that's what this direction said. You can take NAD, add electrons, and form NADH. Once you do, these are broken. Like I've added a chemical that will not allow this to be regenerated back to NAD. So once you run out of your cellular stores of NAD, glycolysis will shut down because it requires the oxidized form, which you just told me is broken. I can't oxidize NADH back to NAD. So glycolysis will stop. Once my stores of NAD are depleted, pyruvate oxidation will stop. And again, once I've exhausted my cellular stores of NAD, Krebs will stop. So let's instead change that to, oh man, I'm just kidding. So sorry. Did I say NADH was broken? What I meant was FADH2 is broken. Oh, well that's real different, isn't it? So you're telling me that my NADH can be recycled back to NAD? Yeah, sorry, my bad. I really was trying to say FADH2. Oh, well cool, glycolysis will still work, right? There's no FADH2 there. Pyruvate oxidation will still work, but Krebs won't, right? Krebs will not be able to cycle because you're going to run out of your cellular stores of FAD, right? And because you're telling me, oh, you're bad. You meant FADH is broken and it can't be metabolized anymore back here. So what about the electron transport chain? Would it keep working? Well, sort of. Because the NAD can be dropped off at complex 1, but your FADH, you said they're broken. So even if they go to complex 2, you said they're broken. So they can't drop their electrons off. So in that case, everything would still work, but you would make less ATP because you would no longer have the 2 FADH2s that would drop their electrons off. And you would stop at some point your Krebs. So initially you would have, everything would work, right, until your FAD stores were depleted. Then only glycolysis and pyruvate oxidation would work. Krebs would no longer work, and your electron transport chain would only be fed by NADH. So do you see the kind of things they can ask you that you'd have to reason through that require you to go, oh, wait a minute. You're saying that my FADH2 are broken, like I can't go back and forth between full and empty and full and empty? Well, then what's going to happen with Krebs? Do not ever say, well, it'll use something else. If I tell you all the food in the world is gone, are you going to go eat wood? No. Okay? It stops. So when you cannot go back and forth between these two forms, whenever the amount of this is gone that your body has, the cell has, stored, it's already got, when it's used up, we've made all of the FAD into FADH2. It's all been reduced by adding electrons. You're done. That's it. Krebs will stop because it is a cycle, and if this is not available, the whole cycle stops. Okay? I hope that helps. So, I'm right at my budget, so I think you're wonderful. We'll do the best we can this week with videos. I might have to load them as I get them done. They take a lot of time to process, but I think you're wonderful. If you need me, you do whatever you need. You email me, whatever, okay? All right, I will see you on Wednesday.